enzymes in chemical reactions. Specifically, the learning objectives are to define the structure and functions of enzymes and to explain their mechanism of action. And then by the end of this video, we should also be able to describe and explain factors that can either increase or decrease the enzyme activity, which determines the chemical reaction rate. So in a previous video, we talked about how some chemical reactions are called irreversible because they have a very large activation energy. This is true, but in our body, most biological reactions are actually reversible. So they can go in both directions. And that's true even if they have large activation energy, and that's because of the actions of the enzymes. So if you remember one thing from this video, is that enzymes actually lower the activation energy necessary for chemical reactions to happen. So we've seen this slide before. And the take-home message from this slide was a comparison of the activation energy. So on the left-hand side, we had low activation energy. So there's just a little hump to get over for this reaction to happen. And on the right, we had this really large activation energy. And we had said that because of this reaction was less likely to happen. Now I want you to focus on what's in the brackets here, is that enzymes can actually facilitate those reactions that have high activation energy. And they do so by actually bringing the activation energy down. So the curve may look more like this with an enzyme, making this chemical reaction more likely to happen. And that's what we see on the graph here, right? So in the absence of the enzyme, you would have this huge activation energy. So you would need a huge push for this reaction to happen. Now with an enzyme, this activation energy is much more reasonable. So not only the reaction is more likely to happen, but it's going to happen much quicker, much faster, because there's less energy required. So what actually is an enzyme? On the right here, we have a cartoon of an enzyme. So in gray, we have the enzyme. And an enzyme is a protein. So it's made of amino acid that are linked together and then folded. So it's a 3D structure. And we'll see how that's important in a few slides. And importantly, on the enzyme, you would find some active sites. So here we have two, one for each reactant or one for each substrate. So one active site here and one active site there. Now it's important to realize that this enzyme is specific to its substrate. Think about it as like a lock and a key. So the enzyme is the lock, and only very specific keys can fit within the active sites because of their very intricate shape. So because of the enzyme structure, the substrate can actually bind to the active site, and that speeds up the chemical reaction. So what exactly does the enzyme do? We keep talking about an enzyme lowers the activation energy, but what does that mean like in real life? So the answer depends on the enzyme. So some enzyme will speed up chemical reactions by bringing both substrate in the right orientation. So they'll basically make sure that the substrates are lined up properly to favor the chemical reaction. Other enzymes will create an environment where it's more likely that the reaction will occur. So for instance, some substrate might like an acidic environment, and the enzyme will be able to provide that. Another way that enzymes can lower the activation energy is by basically bending the substrate. So you bend the substrate a little bit, and that actually makes it easier for those chemical bonds to break. So there's different ways how the enzyme can work, but at the end, all of them end up decreasing the amount of energy that the chemical reaction needs to happen. So this cartoon here is meant to represent how important the specificity of the enzyme is. And we've already mentioned this. The active sites here have a very specific shape. And only a substrate or a family of substrate that can fit very snugly within those active sites will be able to bind to the enzyme. So here in green, we have substrates that are specific to the enzymes. And we can see in step 2 that they're now bound to the enzyme. When this happens, the chemical reaction is more likely to happen. It's going to happen faster. And then these reactants are converted into their products, which are represented here in blue and in yellow. Now, the products don't have a strong affinity with the enzyme, meaning that the products cannot stay bound to the enzyme. So as soon as the products are formed, they are actually released from the enzyme, and this enzyme is now available for more substrate so that we can keep the chemical reaction going. So this characteristic of specificity is something we've discussed before when we talk about protein-mediated transport. Remember when we talked about carrier proteins and channel proteins? We had these 3 characteristics. So that's a slide we've already seen. These transporters were characterized by specificity, competition, and saturation. Now, these transporters were made of proteins. So are enzymes. And because of that, enzymes share similar characteristics that we already talked about with the transporters. Actually, all proteins do. So when you think about enzymes, you should think of them as being specific to substrates. They are also susceptible to competition. So when enzymes can bind more than one substrate, the substrates will compete with one another. We'll see some examples of that. A lot of drugs use these characteristics to their advantage. And they're also susceptible to saturation. So when all of the enzymes present are working at their maximal capacity, then the reaction rate can no longer increase. So these are all characteristics that we've seen before when we talked about transporters, and enzymes just share similar characteristics. So now let's look at what type of factors will affect chemical reaction. So how quickly a chemical reaction occurs is based on the activity of the enzyme. The relationship is pretty straightforward. If you increase the enzyme activity, you increase the rate of chemical reaction. So the next question becomes, what affects enzymatic activity? So it is affected by temperature and pH. So not surprisingly, most enzymes in our body will work best around 37 degrees Celsius, which is our normal body temperature. Similarly, most enzymes will work best at pH around 7.4, which is a neutral pH and the pH of most of our body. So here are a few graphs that illustrate that. On the left here, we see pH, where we have pH on the x-axis and then the rate of enzymatic activity on the right axis. And you can see that if the pH is lower than normal, so more acidic than normal, enzymatic activity decreases. And it's the same thing if you increase pH to a more basic environments, enzymatic activity goes down. So the sweet spot, the optimal pH, is really around 7.4. And you can see the same type of curve for temperature. So we have temperature on the x-axis and rate of enzymatic activity on the y-axis. And you can see that the sweet spot here is around 37 degrees Celsius. Now, why is that? We alluded to the fact that enzymes are proteins with a 3D shape, okay? And that's what we're trying to represent here. So this is the basically 3D structure of hexokinase. It's an enzyme that's specific to glucose. And you can see the active site is right here. And if everything goes well, and hexokinase is in an environment with a pH of 7.4 and a temperature of around 37 degrees Celsius, it stays in its nice 3D shape, and then glucose can bind to the active site and the chemical reaction can proceed. Now, if you stray away from those values, if you put hexokinase in a pH of 5 or if you put exokinase at a temperature of 50 degrees Celsius, for instance, they start losing that 3D shape, which we call denaturation. So the shape of the active site here is lost. And that's so important. Think back of that lock and key model. If you were to change the shape of the lock of your door of your house, well, the key would no longer fit in it and the lock would lose its function. It's the same thing with the enzyme. If they lose their 3D shape, they lose also their function. And that's what happens if you go outside of the pH and outside of the optimal temperature range. In addition to temperature and pH, there's other factors that affect enzymatic activity. Enzyme concentration and substrate concentration are two other important factors. Once again, the relationship should be pretty self-explanatory, where if you increase the concentration of enzyme, you will increase the activity of this enzyme and therefore speed up the reaction rate. Same thing for the substrate. If there's more reactant or if there's more substrate, you increase the activity of the enzyme. This is true up to a certain point, up to saturation. So think about an environment. I'm going to do a beaker here with a certain number of enzymes, which I'm drawing in green now. I'm going to do 3, and a certain amount of substrate. So if there's only 2 pieces of substrate, which only occupy one of the enzyme, adding substrate will speed up the reaction rate, right? We went from 2 pieces of substrate to 6 pieces of substrate, and now we have 3 of the enzymes that are occupied, that are working, therefore we've increased the reaction rate. But what we have now drawn is a situation of saturation. At this point, we have all 3 enzymes that are already working at their maximal capacity. So in this case, adding more substrate would no longer increase the enzymatic activity, would no longer speed up the reaction rate. So this relationship with substrate concentration is true, but only until we reach saturation. And finally, we can modulate enzymatic activity via a number of different modulators, which we're about to describe. So we're going to talk about cofactors, competitive inhibitors, and allosteric modulators. So starting with cofactors. So cofactors are typically an ion like magnesium or calcium. And they are required for some enzymes, but they're not required for all enzymes. And let's focus on the cartoon here, okay? In blue, we have the enzyme, and in purple, we have the cofactor. So for enzymes that require a cofactor, the cofactor is necessary for the creation of the binding site. Another way of saying binding site is active site. So these are actually interchangeable terms. They mean the same thing. So without the cofactor here, there's no active site on the protein. So literally, the enzyme is completely inactive because there's no place for the substrate to bind. Now, when you add the cofactor, like calcium, for instance, it changes the shape of the protein, changes the shape of the enzyme, and now the substrate that we see in yellow here have their active site where they can bind, and now we have an active enzyme. Moving on to competitive inhibitors. These guys block the substrate binding at the binding site. And it's underlined here because it's an important concept that's going to help us differentiate between competitive inhibitor and allosteric modulators, which is the next slide. So once again, let's focus on the cartoon here where we have the enzyme in blue. And you can see for this one, the active sites are available here. Okay, so it's an enzyme that does not require cofactor in this specific case. So normally the substrate or the ligand, same thing here, would bind to this one binding site and this second binding site. When a competitive inhibitor is present, it does what the name says, it competes with the ligand, meaning that its shape mimics one of the substrate so that it can actually fit in the binding site. And we see that on the second cartoon here, right? So you have the inhibitor in green that's bound to the binding site. Therefore, that binding site is no longer available for the original substrate. And as a result of this, the enzymatic activity and the reaction rate decrease. And the name says it. If you're not familiar with inhibitor, to inhibit is to prevent or to block. So whenever you see inhibitor, you should think about it as something preventing a reaction from happening or blocking a reaction from happening. So now let's compare and contrast this with allosteric modulators. So these guys bind the enzyme, but they bind it away from the binding site. And they can either activate the enzyme or inhibit the enzyme. So let's first focus on the left-hand side here. So here we have an allosteric activator. So the allosteric activator is going to increase the activity of the enzyme, therefore increasing the reaction rate. How does it do that? So we have enzymes like this one on the left here that are inactive unless an allosteric activator is present. So they're inactive because the binding sites are not available. Now what an allosteric activator does is that it's going to bind to the protein elsewhere. Okay, it does not bind at the binding site. It binds somewhere else in the protein, right? That's what we see here in purple. So we have the activator that's bound to the protein. What this does is that this induces a conformational change. Remember, this is just a fancy term to say that the shape of the protein changes. And as a result, the binding sites become available. So basically the binding sites were basically like hidden inside the protein here. You put the allosteric activator and those binding sites become available at the surface of the protein, and now the ligand can bind. The reaction rate increases. If we move on to the right side of this cartoon here, we're looking at an allosteric inhibitor. So as we just discussed, this would decrease the reaction rate. So an allosteric inhibitor works very similarly to an activator, but the result is the opposite. So an allosteric inhibitor still binds the enzyme away from the binding site. It will still induce a conformational change. That means that the enzyme will still change its shape, but now we're going from a situation where the binding site was available before, and after the allosteric inhibitor binds, then the binding site is hidden, is no longer available. So to recap, allosteric modulators always bind away from the binding site. Once it binds, it's going to induce a conformational change and either make the binding site available in the case of an activator or unavailable in the case of an inhibitor. So as I mentioned earlier, there's many drugs out there on the market that worked as enzyme inhibitors. So sildenafil, best known as Viagra, is a competitive inhibitor of the enzyme PDE5. So when you decrease the activity of PDE5, you actually prevent the degradation of cyclic GMP, and through a cascade of events that will allow more blood flow to the penis and allow an erection. ACE inhibitors, which are a large class of drugs to treat blood pressure, are also competitive inhibitors. They bind specifically to the binding site of the angiotensin converting enzyme, or ACE, and that prevents blood vessels from becoming narrower. And it decreases blood pressure. So you give that to people who have chronically high blood pressure to help them manage their disease. All right, so just to summarize here, your allosteric modulators, they bind away from the binding site. And they can either increase or decrease the activity of the enzyme, depending if it's an activator or an inhibitor. Competitive inhibitors bind at the binding site. And they always decrease the activity of the enzyme. Cofactors build the binding site, right? They're required for the binding site. So they build the binding site. And they always increase the activity of the enzyme.